Hello, my name is Rachel Bailey, and I'm an assistant professor at UT Southwestern Medical Center, where my lab focuses on the development of gene therapies for neurological disorders. We do this for both rare pediatric disorders as well as age-related neurodegenerative disorders. I am also a scientific advisory board member for the CSNK2A1 Foundation, and I want to thank them for the opportunity to present on gene therapy development for rare disorders. Today, I will be discussing the basics of gene therapy, how gene therapies are developed for rare diseases, and how patient-focused foundations can contribute to this process, including an example of how this has been successfully done, as well as what are potential pitfalls to consider. So what is a gene therapy? As a reminder, genes are made up of DNA and provide the instructions for building the proteins and enzymes that perform crucial bodily functions. A gene variant, also known as a gene mutation, is a small change to the DNA within our genes that can alter the instructions for how proteins are built and work. Gene changes can occur from aging, environmental exposures, be inherited from parents, or can occur de novo in a child where the parents are unaffected. Gene therapy is the introduction, removal, or change of the DNA or RNA within patient cells to target the cause of disease rather than treating disease symptoms. This is done by reducing levels of proteins that can cause disease, increasing the amount of disease-fighting proteins, or by producing new or modified proteins. To develop effective therapies, we must first understand what the molecular basis of the disease is to determine what would be the best therapeutic strategy. To assess disease mechanism, researchers such as myself use cell models, including patient cells, as well as animal models of disease. In devising a strategy, we consider alterations that may occur at the genetic level, if there is a loss of normal function, or a toxic gain of function at the protein level, to then determine if we would need to do gene addition, gene editing, or gene silencing. Once we've decided on a strategy, we then focus on how the therapy is delivered to cells. The new genetic material, such as a working gene, is delivered into a cell using a vector. A vector is like a package used to deliver a specific message. There are many types of vectors, and one of the most common approaches is to use a virus. This is because viruses are very good at entering cells. We can engineer viruses to remove their DNA and use the virus shell as a delivery vehicle for a therapeutic gene. There are many different types of viruses that are used for gene therapy, and each has its own benefits and limitations. How the vector is delivered to patients can be broadly broken down into two categories, in vivo and ex vivo. In vivo delivery is where the genetic material is delivered directly into the person, such as through an injection. Ex vivo treatment removes the patient's own cells and delivers the genetic material to these cells outside the body. The modified cells are then returned to the body. For neurological disorders, such as OCNDS, treating the brain is difficult because of the presence of a blood-brain barrier that prevents most drugs from being able to enter the brain through non-invasive delivery methods. To overcome this, we commonly use a virus called adeno-associated virus, or AAV, because it is very good at discreetly entering the body. We can put a therapeutic gene inside of the virus shell and use it as our delivery truck to transport it safely into the body, where it can get into brain cells and deliver the therapeutic DNA. Here, I want to highlight the number of gene therapies that are approved globally. A little more than half of these are DNA-based and include ex vivo and in vivo approaches. Importantly, there are 6 in vivo AAV-based gene therapies approved worldwide, with 4 of them prescribed for neurological disorders. With the number of clinical trials run thus far and the approval of AAV-based gene therapy products, AAV vectors have emerged as a viable delivery method for human gene therapy. While the use of AAV gene therapy is promising for many disorders, there are challenges to be considered. The first is regarding safety and immunity. People may be naturally infected with AAV over their lifetime, which can cause an immune response and may exclude patients from later receiving an AAV-based therapy. Participating in an AAV clinical trial may also prevent future participation in other trials or from additional types of treatments. Next, there are costs that go into the work that it takes to prepare a clinical trial, and in particular to manufacture AAV and ensure it is carried out at the highest standard in ensuring safety for participants. There is always a chance that the investigational treatment may not work or may have unexpected side effects. Gene therapy can be an alteration for a lifetime, so people should be aware that there could be long-term effects, both good or bad, that are unknown at this time. That is why gene therapies are monitored in person and for years following treatment. So how do we develop novel first-in-human drugs? Here I'm showing the traditional roadmap for the development of a small molecule. For this, there is a pre-discovery phase in which a drug target is identified. Once that is known, there is typically a drug discovery phase in which 5,000 to 10,000 compounds are initially screened to then move hundreds forward for preclinical testing. From this, a small number of lead compounds are identified to move forward for clinical testing. An investigational new drug, or IND document, is then submitted to the FDA to request permission to start a clinical trial. Once approved, the traditional clinical trial design is to have a phase 1 trial that assesses safety in a small number of patients, which, if possible, then moves to phase 2 testing and a greater number of patients for additional safety testing and initial efficacy testing. If promising, a phase 3 trial in a larger number of patients is then initiated to test for efficacy or the benefit of the drug. If a quantifiable benefit is achieved in a modality specific to the disease, then a new drug application can be submitted to the FDA for review and approval. Even after approval, there is a phase 4 in which the benefit and safety of a drug is surveyed post-marketing. Taken together, this process typically takes 10 to 15 years and can cost around $3 billion. When we look at gene therapy development, though, this generally takes significantly less time and money. This is because it starts with a specific therapeutic target and skips the drug discovery phase, so that 1 to 3 gene therapies may be tested preclinically versus hundreds when compared to a small molecule. When it comes to clinical trial testing, important considerations for rare diseases must be made to account for the number of patients that exist and are able to participate in clinical trials. For a rare disease such as OCNDS, it may not be feasible to follow the traditional 1, 2, 3 trial design. As such, the FDA does allow a single registration-enabling clinical trial where safety and efficacy are tested in parallel in a smaller number of patients. For that trial design to be successful, though, the natural course of the disease must be known and outcome measurements and biomarkers identified prior to the start of the trial. These outcome measurements and biomarkers must be quantifiable and associated with disease progression, so that if you then give a potential therapeutic, you can measure the changes in these parameters over time to determine if the drug has halted disease progression and/or restored normal function or healthy levels of biomarkers. For a rare disease, for the phases of drug development to be successful, it necessitates the participation of patients. So why is that? Well, for a drug to be developed for a disease, there must be a need. If there is a large number of individuals affected, then industry will often lead drug development efforts as there is a high financial return for a successful drug. For a rare disease, though, this burden often falls on families with affected children to then raise awareness and provide the initial funding and drive for research. In addition to funding, drug development requires use of either cell or animal models of the disease to test for target engagement and the potential benefit of a drug. For newly identified or rare diseases, these models may not exist. This is where foundations play an important role in helping to identify the best pre-existing models for researchers to use if available, supporting the development of new models as needed, and helping to collect patient cells to develop cell-based models. If preclinical studies are promising, it is then important that advocates help support clinical trial readiness. This comes in the form of determining biomarker and outcome measurements through natural history studies and being able to identify patients eligible to participate in a clinical trial through the patient registry. As part of the regulatory interactions with the FDA, the sponsor of a clinical trial must provide evidence of a patient population that can participate in clinical testing. This is often a challenge for a rare disease, but it's possible by working with a strong and active foundation. So having discussed gene therapy and the development process, I now want to highlight an example from my own work on how a foundation can form these successful partnerships to drive therapy development for a rare disease to enable clinical testing. I began working on a gene therapy for SLC13A5 epilepsy in 2018 after the founder of the Test Research Foundation connected with me while seeking out potential labs that could develop a gene therapy. SLC13A5 epilepsy is a developmental and epileptic encephalopathy where less than 200 patients have been identified worldwide. SLC13A5 epilepsy is a developmental and epileptic encephalopathy that was first described in 2014, when it was first discovered that variants in the SLC13A5 gene are causative of SLC13A5 epilepsy. It was then in 2015 when the first patient-focused foundation was started by the mother of 2 affected children. The goals of the Test Research Foundation are to provide support to affected patients and families, drive research on SLC13A5, develop treatments, and spread awareness. So why was the foundation formed? In 2015, there were less than 10 children diagnosed with this disorder. There were no funds available to research SLC13A5 epilepsy, and there was not a patient registry. There were no advocacy organizations for this disease, and there had been no research conferences to spread awareness and gain knowledge on SLC13A5 epilepsy. At the time, it was incredibly challenging for patients to get diagnosed as genetic testing was not accessible, and there were no predictive biomarkers. In terms of resources, there were 2 disease models available. However, there were no treatment guidelines. Why loss of SLC13A5 caused epilepsy was unknown. There were no disease-modifying treatments, and there were no drugs in development. What was known, however, is that variants in SLC13A5 can lead to an imbalance of citrate levels in the brain. Normally, the protein made by SLC13A5, called NACT, helps move citrate from outside of a cell to inside of a cell. Once inside the cell, this citrate is broken to metabolites and neurotransmitters, which are critical for normal brain function. When there are variants, the NACT does not work properly, and citrate accumulates outside the cell, leading to not enough citrate and potentially downstream products within a cell. Babies born without properly working SLC13A5 genes begin to have seizures within the first week of life. These seizures persist and in some patients occur hundreds of times a day. This leads to developmental delay, including issues with being able to talk and issues with walking and movement. Currently, there are no treatments that address the underlying cause of the disease, and medications to halt the seizures do not work in all patients. Importantly, children with this disease do not gain independence, even as adults, and they can die from the severity of their seizures. The Test Foundation provided the initial funding for my lab to develop a novel vector to treat SLC13A5 epilepsy. With their support, my lab developed a vector to deliver a working copy of the SLC13A5 gene to patient cells, so that their cells could make the protein that is needed and restore normal function. We initially tested our drug in cell culture systems for functionality, and here I am showing SLC13A5 in red being properly expressed and located in the plasma membrane that surrounds the cell in green. I also relied upon the Test Foundation for finding models of SLC13A5 epilepsy that I could use for proof of concept safety and efficacy studies. To test if our gene therapy could be beneficial to patients, we used a mouse where the SLC13A5 genes had been removed from it to mimic the non-functioning SLC13A5 gene found in patients with the disease. After delivering the virus to the mice, we collected blood from the animals to check for citrate levels. Without treatment, we see as expected that mice missing the gene have too much citrate outside the cell. With gene therapy treatment, though, we found that we are able to restore normal citrate levels. We then wanted to know if this treatment has a functional benefit for animals. Using EEG, we are able to measure the brain activity of mice, and here I'm showing an example of normal activity in an unaffected mouse. In a mouse with the SLC13A5 gene is missing, we see atypical epileptic discharges, as shown by the taller blue spikes, while in a mouse that was missing the gene and received the gene therapy, we see that normal brain activity is restored. We then assess seizures in these mice. With repeated injections of a chemical called PTZ, we can cause seizures that become more frequent and severe with each injection. The black line shows a low seizure severity score in a normal mouse, while the blue line shows a high seizure severity score in a mouse that is missing the SLC13A5 gene. When we use our gene therapy in the SLC13A5 mouse, we find that we are able to restore the seizure severity to healthy levels, as shown by the orange line. With severe seizures, there is a greater risk of seizure-related deaths. As shown by the blue line, a greater number of mice missing the SLC13A5 gene die from the severe seizures. When we give the gene therapy, though, we see that these mice are protected from seizure-related deaths, as shown by the orange line. Together, this data, and other data that I'm not showing for the sake of time, supports that our treatment could provide benefits to patients with SLC13A5 epilepsy. With that data, we were then able to identify an industry partner that was willing to fund the pivotal dosing and efficacy studies, and to then perform rigorous toxicology studies in larger animals. To find an industry partner, it was important to include the Test Foundation, as they provided the critical patient component that is needed for a successful clinical trial. This comes in the form of identifying biomarker and outcome measurements through natural history studies, and being able to identify patients eligible to participate in a clinical trial through the patient registry. I want to reiterate that with the drive and resources from the Test Foundation, we were able to combine gene therapy research data with knowledge of disease progression and patient treatment from our clinical collaborators, and using therapeutic development resources from an industry partner to develop a drug specific for SLC13A5 patients. And importantly, it took all of these partnerships for success. So where are we now? Looking at the roadmap for developing a gene therapy, with the support from the Test Foundation and industry sponsor, we have developed a novel gene therapy for SLC13A5 and completed the preclinical testing, which supports moving this drug forward to the clinic. As such, we have received guidance from regulatory agencies through pre-IND meetings on how to proceed. The Test Foundation is supporting an ongoing natural history study at Stanford University, Brown University, and UT Southwestern for us to identify biomarkers of disease and outcome measurements. Using this information, we are moving towards getting regulatory approval to start a clinical trial, and this is being done as a partnership. So to bring it back to the Test Foundation slide and to highlight the progress they've made. In 2024, there are now 129 children diagnosed with disorder, and $10 million have been obtained to support SLC 13A5 epilepsy research, and there is now a patient registry. The Test Foundation is the sole advocacy organization for SLC 13A5, and they have held 7 research conferences since their inception. Genetic testing is now available through an epilepsy panel, and citrate has been identified as a potential predictive biomarker. There are over 14 different cell lines and models for SLC 13A5 research, and treatment guidelines are in progress. Why a lack of citrate transport causes epilepsy is still unknown, and there is not an approved disease-modifying therapy yet. However, with the drive and resources from the Test Foundation, there is now a gene therapy drug under development. While I hope I have shown how a foundation can work with academic and industry partners for rare disease drug development, I do want to highlight potential pitfalls in these relationships. These largely lie around mismatched expectations in regards to timelines, reporting, data sharing, and ownership, as well as outcomes. With pediatric diseases, there is an urgency for results and program progress that may not be realistic from an academic or industry standpoint. From the academic side, delays in research can occur from a limit of resources, including underfunding of a project, or delays that occur at the institutional level in terms of contracts and agreements. It's important to note that while academic partnerships are more affordable than, say, contract research organizations, this is largely in part because labs and PIs have multiple projects and duties they must accomplish to maintain their funding and position. From the industry side, mismatched timelines can arise from similar issues such as limited resources or delays in manufacturing. Importantly, though, while industry has the potential to move quickly and with many resources, the ability to do so is based on program prioritization and what the risk versus benefit proceeding with the program is. As such, where there have been cases of successful partnerships of foundations with academics and industry, these have been because there are clearly defined and agreed upon roles. This includes having frequent and open discussions on expectations and timelines as drug development is an ever-changing landscape. With that, I will end on this slide. I'd like to acknowledge the former and current people in my lab who have worked hard on the SLC 13A5 gene therapy program that I discussed today. I'd like to also thank my natural history study collaborators, Dr. Brenda Porter, Dr. Kimberly Goodspeed, and Dr. Judy Liu for providing scientific and clinical expertise on SLC 13A5. And I am grateful to Kim Nye and the Test Foundation who have supported this research from the beginning and are working tirelessly to fund research and treatments for SLC 13A5 deficiency. So thank you again to the CSNK2A1 Foundation for the opportunity to speak and also for your attention.